Well, hello my fellow pre-meds and welcome back to another video on lab techniques. We're specifically going to be covering electrophoresis, immunoassays, and blotting. All lab techniques that kicked my butt in my neurology research project. But luckily for us, learning about these lab techniques is a lot easier than applying them in the lab. Also, there are a lot of subcategories within these broad categories, so if you wanted to jump to a specific lab technique, you're able to do that. I've time-stamped this video. Let us get started with electrophoresis. So the purpose of gel electrophoresis is to separate molecules based on size, shape, and charge. Although, as we'll see, what really ends up mattering on the MCAT in the majority of cases is size and size only. And we'll talk about that later in subsequent slides. Now, let's familiarize ourselves with the gel over here. You can see that you have a negative cathode and a positive anode. So I know in Gen Chem, we typically think of the cathode, especially with galvanic cells, the cathode as the positively charged electrode. In this case, the cathode is negatively charged. Okay, so when it comes to size, the smaller the molecule, the farther it's going to travel. You know, basic physics, force equals mass times acceleration. The smaller the mass, the bigger the acceleration. That's about as far as we'll go as it pertains to physics in this video. Next, we have charge. The more negatively charged a molecule is, the faster it'll travel because it's more attracted towards that anode, that positively charged anode. And finally, shape. The more compact the molecule is, the faster it'll travel. There are two gels that we can possibly use. So when it comes to DNA and RNA, we're going to use agarose gels because they have larger pores and DNA fragments are larger. And then when it comes to protein, we want smaller pores, so we're going to use polyacrylamide. And also a few more things regarding the gel. As you can see right here, you have multiple lanes. That first lane is typically what we call the ladder lane. We use it as reference. So it allows us to infer what the weight of our molecule, the molecular weight of our molecules are. So let's say in sample one over here, you can see that you have a band right here. You can infer that whatever molecule is present here is the same molecular weight as this molecule, and therefore, and we already know this before because we designed it, we know that it is 6,000 Daltons, and so on and so forth. Now, when it comes to the thickness of the band, you can see that this band is thicker than the rest. That has nothing to do with the molecular weight. It just tells you the relative abundance. So we probably have more concentration of that specific protein relative to the other ones, and that's why the band is thicker. Okay, now let's talk about why only size really ends up mattering. First, we're going to talk about DNA and RNA. So we know that DNA and RNA are made of DNA is double helix and RNA is single-stranded. In either case, their backbone is made up of phosphate sugar molecules, and the phosphates carry a negative charge. And therefore, the overall charge on DNA and RNA is going to be negative, and therefore charge really doesn't matter when it comes to DNA and RNA. Likewise, shape doesn't really matter because they're both like either double-stranded or single-stranded. So when you're comparing DNA to DNA, same shape, RNA to RNA, same shape. Now I know what you might be thinking. What if we have a larger DNA fragment? Wouldn't it carry a bigger negative charge? Well, it turns out that extra speed it's going to gain because it contains a negative charge, so it's going to travel farther technically if it has an extra negative charges, right? Because it's going to be attracted towards that positive electrode. It turns out that extra speed is offset by the speed it's going to decrease because of its increasing size. So they kind of cancel each other out. So that's why when it comes to DNA and RNA, only size matters. But how about for proteins? We know that proteins come in all different shapes, right? And they can carry all different types of charges. Like they can have an abundance of amino acids that carry negative charges versus positive charges. They might have a mix of both. So why doesn't charge really matter or shape really matter? So the reason is because we use this specific technique called SDS page. The long name for that is sodium dodecyl sulfate polyacrylamide gel electrophoresis. All this is saying is that we're using polyacrylamide gel, that's that page part of it, and then SDS, a specific detergent that we use, it's going to unfold your protein and coat it with an overall negative charge. And so what ends up happening to these proteins when you add SDS is they linearize, so you can see that over there. So now they have roughly the same shape, and they're coated with negative charges regardless of what kind of amino acids they had before, whether they were positive or negative. So that's why when you apply SDS page, size only matters, charge and shape do not matter. If you don't use SDS page, so instead you use negative page where you don't apply that SDS detergent, then in that case, indeed, shape, size, and charge will all matter. Okay, now let's move on to an offshoot of electrophoresis, which is isoelectric focusing. The purpose of this is to determine the PI of proteins. So just a quick review behind the concept of a PI or isoelectric point. So we go in depth on this topic in our biochemistry playlist on the PI of amino acids. Here we're going to do a quick review. So the PI of neutral amino acids is going to be about 6, the PI of acidic amino acids is about 3, and the PI of basic amino acids is about 10. The PI is when the molecule, the amino acid, exists as a zwitterion, so its overall charge is 0. And we can see how we get that based on these diagrams over here. So you can see with a neutral amino acid, you're going to look at the backbone groups, so the backbone amino group and the backbone carboxyl group. Take the average of those pKa's, and that is your PI. So it's about 6. And then for acidic amino acids, you're going to take the average of the backbone carboxyl group and the pKa of your side chain. Okay, so because it's going to contain a carboxyl group also in its side chain, so you're going to average those to get your PI. And then finally, for basic amino acids, you're going to take the average of the pKa of the amino group in the backbone and the pKa of the amino group in the side chain. So that was just a quick review on how you determine the PI values over here. But it turns out that only acidic and basic amino acids will contribute to the overall PI of a protein. And that's because, think about like where the carboxyl group and the amino group that are neutral amino acids, where they end up being in the protein. So you can see that this, for example, is a neutral amino acid. This one is also a neutral amino acid. Their groups that are capable of carrying a positive or negative charge are embedded in these peptide bonds, right? Because the peptide bond is formed by joining this carboxyl group with this amino group. So it's embedded over here. So now they can't contribute to the overall charge of a protein. The only thing that can is side chain amino acids that can carry positive or negative charges because they're not embedded within those peptide bonds. Okay, so now with that quick review in mind, let's start talking about the isoelectric focusing technique. So we're going to apply it to amino acids, and then we're going to infer from there how it works for proteins. So you start off with this gel, again, positive electrode, negative electrode, and then you apply a pH gradient that goes from 0 to 14. Importantly, 0 should start at the positive electrode, and then you should have 14 at the negative electrode. So what you're going to do is you're going to apply your samples on either side. We're going to start on the positive side. And we have 3 amino acids. The first one is our neutral amino acid, second one is acidic, and third one is basic. Notice that right now they all carry positive charges, and that makes sense because at low pHs, let's say a pH of 1, the surrounding environment is acting as an acid, thus donating protons to every single group on these amino acids, thus giving them all an overall positive charge. So we can see that our neutral and acidic amino acid carry positive 1, whereas our basic amino acids carries positive 2 because its side chain is capable of carrying another positive charge. Okay, so what's going to happen now is we apply the current, and the amino acids are going to start to migrate towards the negative electrode, right? Because they currently have a positive charge. They're going to keep on migrating until they carry an overall neutral charge, right? When is that going to happen? Well, that's going to happen when, for example, for the acidic amino acid at a pH around 3, when its carboxyl group pKa starts to become less than the surrounding environment. So at that point, the carboxyl group donates its proton. Now it carries a negative charge, so it has both negative and positive charge, so it's neutral, and it stops migrating. At that point, when it stops migrating, you know that you've hit your PI for that amino acid. Now notice what happens with the neutral, the one that originated as a neutral amino acid over here. You can see that it continues to migrate until it hits around 6, right? Because the PI of a neutral amino acid is around 6. So that's where it's going to stop, at its PI. And then look at the basic amino acid. Well, it continues to travel all the way down to about a pH of 10, a little over that, because that is its PI, and it won't stop until it hits that PI value. Now, one point of confusion may arise because these look identical, even though I said this one's neutral and this one's acidic. Well, the idea is that over here, the side chain, this contains a carboxylic acid group, but at a pH of 1, it's going to be protonated, so it's not going to carry a positive or negative charge, and therefore they just appear similar. They appear identical. Okay, so this is just reversing the process. So we started at the positive electrode, but we could have easily started at the negative electrode, right? Where the pH is 14. If I put my amino acids at a pH of 14, well, now the surrounding environment is acting as a base, thus taking away all the protons from my amino acids, all the groups of my amino acids. So regardless of whether it's acidic, basic, or neutral, they're going to carry a negative charge. And therefore, they're going to start migrating towards the positively charged electrode, and they're going to stop at their PI values. So it doesn't matter where you started from, they're going to stop migrating once they hit their PI value. Now let's apply this to proteins. So over here, we just combined both the diagrams we saw earlier where we first did positive side and then the negative side. Here we did them together. So we put a bunch of proteins on the positive side, a bunch of proteins on the negative side. Of course, the ones on the positive side will carry a positive charge because you have a lower pH, protonating your samples, giving them positive charge. And on that side, they'll carry negative charges. And then you allow the current to run. The proteins will start to migrate in either direction. They won't stop until they hit their PI values, at which point they carry an overall neutral charge. So for example, here they can ask me, what is the PI of this green protein? I look at where it stops around 5, so 5, and you can see it over here, 5.6. So you can infer based on that what the PI value is. Another question they can give you is, what do you think is most abundant? What type of amino acids are most abundant in this protein? Well, you probably have more acidic amino acids in this protein compared to basic ones, and that's why its PI was on the lower side. Okay, so now let's go over blotting. So blotting is sort of the next step after electrophoresis. So electrophoresis, the goal of it was to separate your molecules. Now the purpose of blotting is to be able to identify what specific molecule you have. So let's say I had a sample that contains insulin, but I didn't know that, and I wanted to identify whether I have insulin or not. I can use blotting to identify that. Or, for example, if I wanted to apply it to DNA, I can try to see if I have the specific fragment that's part of the gene that encodes for insulin. So first, we're going to talk about blotting in general terms, how it works for proteins, DNA, and RNA, and then we're going to break off and talk about Western, Southern, and Northern blot for protein, DNA, and RNA, respectively. So the general workflow is that after you separate your molecules by electrophoresis, that's what we see over here, we've separated them by electrophoresis, you're going to blot your gel, okay? So blotting just means like to transfer your gel onto a membrane. So this is your gel over here. You're going to transfer it onto a membrane. By the way, this part of it is so annoying. You literally have to add like 10 layers and make sure that they're all added in the correct order. As you can see right here, like the filter paper, the gel, the membrane, the filter paper, the sponge on top. Like this was really annoying when I was working on my neurology research project. But anyway, so you just transfer the gel onto the membrane. And now you can see right here, this is when we finished the process of blotting. So now we have our membrane, which contains our samples, okay? So what you do next is you want to add a labeled probe, and we'll see what this probe is exactly in the different cases and the different blotting techniques. But essentially the probe is going to light up so that if your protein of interest, your DNA fragment, your RNA fragment of interest is present, it will light up and you'll be able to detect it. So it'll tell you whether that protein is present or not, and it'll tell you the relative abundance or the relative concentration compared to the other samples of how much is present in your sample of interest. But it's not going to tell you the exact concentration. So like if we take a quick look over here, once we've added the probe over here, you can see that it lit up in lane 1 over here and lane 3. So from here we can say that lane 1 and 3 both contain insulin, or in the case of DNA, the insulin encoding gene, and we can infer that this specific band, this specific sample contains a higher concentration relative to this one because that band is thicker. But importantly, we cannot figure out the exact concentration. So now let's talk about the different kinds of blotting. We're going to start off with Western blotting. I know it's kind of customary to start with Southern blotting that's specific to DNA, but I feel like this one's a bit more straightforward, so we'll introduce the idea with Western blotting and then we'll break off to, or branch off to, Southern and Northern blotting. So the idea here is, first you run your gel, again, we're using SDS page, so we're linearizing our proteins and then running the gel, and then we blot, which we just saw earlier, so you blot onto your membrane, and then you add your probe, and you can see that it lit up indeed here and here, and this is the image we saw previously. The only difference here is that we're going to discuss what specific probe you're using for Western blotting. So the probe is going to be an enzyme labeled antibody. So essentially what's going on here is that when you add this antibody to this blot, it's going to go and bind to your protein of interest. That is, if your protein of interest is present. So if it's present, it's going to bind to it, and then you're going to allow a reaction to run using this enzyme, and then you'll get like a fluorescence. You'll be able to detect a signal. And sometimes you don't even need an enzyme. You can just have an antibody labeled with some like fluorescence material that'll light up. So if you see that light, then you know that that specific sample contains your protein of interest. In this case, let's say it's insulin, for example. Of course, this antibody would be specific only to insulin. So if insulin weren't present in any of these samples, you would see no light and you'd know that you don't have your protein of interest in the sample. So importantly, once again, this is going to identify whether the protein is present or not and its relative abundance. And as we discussed earlier, this one will have a higher concentration compared to this one because it's a thicker band. Importantly, they should be at the same level because they're the same molecule, so they should be at the same weight, here about 8,000 Daltons. Okay, let's move on to southern blotting. It's a bit more complex, but still overall pretty straightforward. So first you need to cut your DNA fragments. So let's say you get 3 fragments of DNA and you wanted to see which one contains a specific sequence that's part of the insulin encoding gene, for example. So first thing you have to do is use restriction enzymes, which we call molecular scissors, because that's exactly what they are. They're scissors that cut up DNA. We'll discuss them further in the next video in this playlist on biotechnology. But for the purposes of this video, just know that they cut up DNA at specific restriction sites. So you can see that they'll cut up your larger fragments into smaller fragments. And then after that, kind of what we did earlier, you're going to run your gel. Here, I just wanted to give you like your fragment of interest. Let's say your fragment of interest is 5' A, A, C, C, T, T, etc. So we're going to assume that your fragment of interest is present in one of these, okay? And that's why we're doing this whole technique, to find out if it is indeed present here. So let's say this is a sequence, part of a sequence from the insulin encoding gene. All right, so now you want to perform agarose gel electrophoresis, then blot. So that's what we did over here. Now this membrane over here, after you've blotted, contains these double-stranded DNA fragments, right? So what you have to do, there's also an additional step over here with southern blot, is that you have to denature these samples. Because recall, we're looking for a specific sequence over here. So we have to denature the samples so that rather than them being double-stranded, they are single-stranded. That's what we do over here. You can see right here, denature your fragments using NaOH, a strong base. So it's just going to separate the double-stranded DNA molecules into single-stranded ones. Okay, so now essentially what you did is you bathed this membrane, you put this membrane in an NaOH solution to allow it to denature. So now these bands consist of single-stranded DNA. Now, and only now, can you add your probe. Well, and again, that's your fragment of interest over there. What is your probe? The probe is a fluorescence-labeled single-stranded DNA molecule that you previously designed that contains the exact sequence that is complementary to your sequence of interest. So if you want to take a look at what that's going to look like, it looks exactly like this. It's a single-stranded fragment that's fluorescently labeled so that you can see if it lights up to identify the specific band that contains it. And then it's going to be complementary to that. So notice 5' AACC, 3' TTGG, and so on. Okay, so now we put the probe onto our membrane, and what you're going to notice is if it's present in your membrane, it's going to light up, and it indeed lights up in lane 1 and lane 2, and therefore lane 1 and lane 2 both contain our sequence of interest. And importantly, they contain it in equal concentrations because, as you can see, the bands are of the same thickness. Okay, so this allowed us, once again, I really want to hammer this point home, this allowed us to identify whether a DNA sequence is present or not, and its relative abundance, so its relative concentration. So if one band was thicker than the other, then we would know that the thicker one contains a higher concentration of that DNA sequence. Next, northern blotting. We're not going to discuss it in detail because it's pretty much the same exact process as southern blotting. Really, the only difference is that you don't need to denature your sample because this is already single-stranded RNA, right? RNA is by definition single-stranded, so you don't need to denature it. So you're going to cut your samples, you're going to run the gel electrophoresis, and then you're going to blot and add your probe, which again is single-stranded DNA that's fluorescently labeled so that you can detect whether your RNA sequence is present or not. So here are some questions. I don't typically like to include questions in these slide decks, but I do throw them in every now and then. I will have separate videos where I go over specific videos for questions and exercises on these specific topics. But let's go over this real quick. So identify the probe type used for each blotting technique. For southern blotting, we're trying to detect DNA, so single-stranded DNA. For northern blotting, we're trying to detect RNA, so again, single-stranded DNA. And then for western blotting, we're trying to detect a protein, so it's going to be an antibody. All right, now next question, which blotting technique techniques can be used to detect the level of expression of a gene. So when you think gene, you might automatically think DNA and therefore choose southern blotting, but that would be wrong because that's only telling you that that gene is present. Whether it's expressed or not, it doesn't really tell you much information on that. You need to use either western blotting to see if the proteins are being translated, or northern blotting to see if mRNA transcript are being transcribed to determine whether that gene is being expressed or not. So the answer here is western and northern. Okay, so this last scenario shows up a ton on the MCAT. Of course, not the exact same scenario, but the idea behind it. Insulin is composed of two polypeptide chains connected by disulfide bonds. After electrophoresis and western blot analysis of two samples known to contain insulin, a researcher observes the following results. What best explains the findings? So notice the researcher knows for sure, and let's assume in this case that only insulin is present in the two samples. But when they ran the electrophoresis, they noticed that one of the lanes contains one band while the other lane contains two bands. So what best explains this? Well, it probably makes sense, especially since these two bands appear to be lighter than this one, right? Because they traveled farther. Then it probably makes sense that maybe the insulin that was originally in this lane was broken down for some reason, and that's indeed what happened, because as they mentioned, there are disulfide bonds joining the subunits to one another. So maybe, as we can see right here, you had a reducing agent. So you can see when you have disulfide bonds, you can add a reducing agent to break them down, and then you separate the subunits, and that explains why you have two separate bands over here. Essentially the insulin broken down into two separate parts. And over here, the insulin was preserved, and therefore it's heavier, because obviously insulin can be only heavier than the individual components that make it. All right, let's conclude this video with a relatively easy to digest topic relative to the previous ones, specifically immunoassays, and we're going to cover two of them. The first one, enzyme-linked immunosorbitant assay, ELISA for short. Now, importantly, these are not very high yield on the MCAT like blotting and electrophoresis, but they do show up, so let's go over them. You have three different types. You have the direct, indirect, and as we'll see on the next slide, we have the sandwich type. And this is how they work. So in the direct one, you have your proteins in this container, in this beaker, and let's say you don't know what the concentration of those proteins are, so you run an ELISA to determine the concentration. The way you do this is you first add an enzyme labeled antibody, which will look like this. So you can see right here you have your antibodies that are specific to this protein, and let's assume this protein is insulin. I don't know why we're using insulin for every example today, but let's just say this protein is insulin. So this antibody will specifically bind insulin. And notice the antibody is labeled with this green label over here. That is an enzyme. And we'll see why it has to have an enzyme on it in a bit. So the next step after that is going to be a wash-off period. You're going to wash any extra antibodies. The idea here is that notice how you don't know what the concentration of protein is over here. So when you add antibodies, you're probably going to add extra antibodies, more than the proteins you have in the sample. And as we'll see, the antibodies and the enzymes on them are used to determine the concentration. So if you have too much antibodies, you're going to overestimate your concentration. And that's what you have. You see over here, we have antibodies that haven't bound to any of the insulin molecules. So you do a wash-off period where you wash off the extra antibodies, and what you're left with is only the antibodies that are attached to your protein of interest over here. So next, what you're going to do is you're going to add the substrate of the enzyme. So the enzyme substrate, which is going to be a colorless sample, a colorless molecule. And then when the reaction runs, you'll notice that what happens is it generates another molecule that you can detect. It'll generate some sort of signal that you can detect. And the higher that signal, obviously, the higher concentration of protein in your sample. So with direct ELISA, the idea is pretty simple. You're using an antibody that's labeled with an enzyme, and then you run a reaction, and based on the product of that reaction, you can determine the exact concentration, not the relative concentration as we saw with electrophoresis, but the exact concentration of insulin or of the protein in your sample. Indirect ELISA just adds one extra step. So you'll see here, first we have this first antibody, which is not labeled with the enzyme, and that's what we call the primary antibody. And then you add the secondary antibody, which is going to bind to the primary antibody in a 1-to-1 ratio. And then based on that, you can determine, based on the concentration of the product over there, you can determine the concentration of your protein. Next, we have the sandwich form of ELISA. So also very similar. Over here, you're coating your beaker with the antibody, and then you're adding your sample and then your secondary antibody will bind to the antibody-antigen complex, and again, you'll run your reaction and determine the concentration. One thing I wanted to mention here, because they do expect you to know some, experimental design, control versus experimental on the MCAT. So I thought it was worth mentioning this over here. While doing the washout period, you'll typically have like a control for that washout period. So the idea is that when you add your antibodies over here, if you have additional antibodies that aren't bound to protein, you're going to overestimate your concentration. And that's why we did the washout period, correct? All right, well, what we do over here is we have this empty beaker, let's say a beaker that doesn't contain our protein of interest. We add the antibody. Of course, the antibody would only bind to our protein of interest. So in the best case scenario, once we wash it off, we should not have any antibodies because we didn't have the protein of interest, so the antibodies are not going to bind to it. So when I try to detect the signal in this beaker, I should not see a signal because it shouldn't have the antibodies after I washed it off. If I do see a signal, then that tells me there was probably an issue with the way I washed off my sample, and therefore the concentration that I measure here is probably not accurate. Okay, so that's why we have a control over there. Okay, let's move on to radioimmunoassay. This one's very straightforward. So the idea is over here we have our beaker or our flask, and then we coat it with antibodies, okay? We coat it with antibodies specific to, again, insulin, the protein of interest for us. So these antibodies will specifically only bind insulin, and then you add radio-labeled insulin. So this insulin has been labeled with a specific label. This isn't the one, let's say we're trying to measure insulin in a patient's blood sample. This isn't from the patient. This is insulin that you have in the lab that you labeled previously for this specific experiment. And so when you add it to the antibodies over here, the insulin is going to be taken up by those antibodies, and you're going to have these antigen-antibody complexes. Importantly, note that they are all bound to this radio label. So they all have a radio label. Okay, so if I were to detect this radio label, the signal will be high because they all have that label. Now, next what you're going to do is you're going to add the patient's blood sample or the patient's sample. And you can see that's what we're doing over here. So we're adding these. Again, the patient's sample contains insulin, but it's not labeled, right? It's not radio labeled because it's the natural insulin occurring in the body. What you'll notice is that the radioactive signal decreases. Why is that? Well, because some of the radioactively labeled insulin molecules, proteins, have been displaced from the antibodies by the patient's insulin samples. And the higher the concentration of the patient's insulin, the more radio-labeled insulin will be displaced, and therefore you will have a lower signal. So what does this mean? This means there's an inverse relationship between the radioactive signal and the insulin concentration. So when you're doing radioimmunoassay, the higher your signal, if they tell you that the signal is higher on the MCAT, then you know you have a lower concentration in solution, and vice versa. If it's a lower signal, you have higher concentration in solution. And this is just reiterating on the point that ELISA and radioimmunoassay are both used to determine the exact concentration of the protein in solution and not just like the relative abundance like we saw with the blotting techniques. Importantly, radioimmunoassay is more sensitive than ELISA. So why would you use radioimmunoassay over ELISA? Because it's more sensitive, meaning even at very low concentrations, it's going to be able to detect the concentration, whereas ELISA may be inaccurate at very low concentrations. Okay, so I just really wanted to hammer that point down. So notice now we're adding a ton of extra insulin molecules. So let's assume that there was a higher concentration of insulin in the patient's blood. You can see you've displaced more of those radio-labeled insulin molecules. Okay, so this is a summary table, very, very high yield. You can screenshot it. You can access the slide deck later and annotate it. And it's also going to be covered in our Anki cards, but it's very, very high yield. So make sure you understand this upside down and backwards. That concludes our video on electrophoresis, immunoassay, and blotting techniques. In the next video, we will discuss biotechnology, the last video of our lab techniques playlist. If you like the way that I teach, check out the rest of the videos in this playlist, subscribe, and turn on the notification bell so you stay up to date with all of my future playlists. And if you have a friend who's currently studying for the MCAT, don't gatekeep these videos, share the videos with them. It really helps the channel grow, and I would really appreciate it. Also, did you know that every single video in my playlist is associated with Anki cards, the slide decks in the videos, and high yield sheets? If you want to access these to be able to better actively review review the material as you're learning it from the videos, then check out my Patreon or reach out to me directly for alternative ways to access those materials. Finally, these videos, at least as of now, are sponsored by you. If you wanted to support me via Buy Me a Coffee or Patreon, you can find the links in the description box as well as the links for everything else. Thanks for watching, and I'll see you in the next video.